The Active Data Biology application has multiple views of datasets, each providing a different set of interactions for users to visualize patterns and browse their data. The Canvas view allows users to investigate and interact with the data they have pinned from all across the site. Each user in a collaborative project has their own canvas, so you can customize and make it your own. In this tutorial, we will cover basic Canvas controls, recommendation cards, and collaborating with the canvas. The dataset we'll use is the User Study Dataset, a publicly available ovarian cancer data which can be accessed with the guest login. We'll begin with basic definitions in Canvas navigation. For the purposes of this tutorial, a pin refers to the circular objects, mostly green, created by you while using the Active Data Biology application. Recommendation cards are the gray rectangles that contain information related to a pin and are automatically generated by searching external knowledge sources. To help organize your space, any pin can be moved around the canvas by clicking and dragging it. The visible screen can be panned, allowing you to move things into or out of view. Scrolling the mouse wheel allows you to zoom in or out. Note that the canvas is much larger than what is visible at the default zoom level. Additional information about navigation can be found in the legend available through the Help menu in the upper right corner. Pins show data that you have chosen to save. For demonstration purposes, we'll start with two different types of pins on the canvas, an individual protein and a protein group. The most common way to create a pin is from another view of Active Data Biology. In both the Heatmap and Pathway view, a pin icon is displayed with various data. Clicking this puts the data onto your canvas. A second way to create pins takes data from PubMed directly to your canvas using our Pin to Canvas bookmark, found on the FAQ page. When viewing a single publication, a pin is created for that exact publication. When viewing the results of a search, a pin is created for the search query. When viewing a single protein, a pin is created for that protein. A third method is to create a custom pin by double-clicking an empty spot on the canvas. Although custom pins don't create recommendation cards by default, they can store certain recommendations created by other pins. Custom pins that start with a hashtag are used to label and group other pins. These are colored differently than normal pins, and any pin dragged onto this group pin will change color to match it. To remove pins from the canvas, hold Delete and click on it. Recommendations gather information from external data sources for users and are created by a proactive search and updated anytime you view the canvas. The front of the recommendation card has a brief description. Flip it over to the back side that contains more information and a link to the external data. Click anywhere on the back of the card to flip it back over. When you have found a useful recommendation, it can be saved by dragging the card onto the pin that spawned it, hiding it from view until it is needed again. To reveal a saved recommendation, click the new icon created on the pin and then click the name of the card. To temporarily hide a recommendation card, hold H and click. This card will be regenerated when the canvas is reloaded. To permanently dismiss a recommendation card, hold Delete when clicking on it. Now we'll show the specific recommendations generated for a few types of pins. We anticipate updating these as Active Data Biology matures, but this snapshot is still instructive. Pinned individual proteins have two recommendation cards. The first card is the protein's data at NCBI. The front shows the protein's name, and the back shows a description of the protein alongside its accession number and the organism. There are links to download a FASTA file, or visit the NCBI webpage to see the full information. The second type of recommendation shows the results of a search for PubMed articles containing the protein's name or accession. Clicking the icon in the lower left of this card displays a few different views of the PubMed search, like a timeline and a word cloud. Flipping the card provides the exact query to PubMed and a link to the search. Clicking on any article opens a recommendation card for the article. Protein groups also have two recommendation cards, the data card and the pathways card. The data card shows the number of proteins and pathways within the node. On the back of the card is a link to download the actual data from the expression matrix for these proteins. The pathways card shows a list of enriched pathways found in this group of proteins. Flipping the pathways card allows the user to scroll through the whole list, and in either state, the user can click on the name of a KEGG pathway to open it in the pathway view. Double-clicking on the pin itself opens the Heatmap view with that node selected. The canvas is designed to be collaborative. To make this happen, we save each person's canvas in a central repository called GitHub. You don't have to learn anything specific about GitHub to use the collaborative functionality, though. We've taken care of all that for you. First, the canvas is saved periodically as nodes on the canvas are updated or if you leave your browser session. You can also initiate a save event by clicking on the canvas icon in the upper right and selecting Save. Likewise, you can reload the most recently uploaded version of the canvas by selecting Update. The best part about collaborating is seeing what others have done. Select the collaborator's canvas to load it onto your screen. If you see something that you like, you can copy a pin by holding C and clicking. When viewing someone else's canvas, we don't allow you to make changes, but you still have the ability to pan and zoom the screen. Although this user's canvas is open, you still pin to your canvas from the heatmap or pathway views. Once you're finished, you can save and reload your canvas by clicking Save and Return to Canvas. The canvas helps keep track of the ideas you have and the progress you make while investigating them. Together with the rest of the Active Data Biology toolset, it will enable you to interact with and explore your data in new ways.